Porcia. I'm from COA, College of the Atlantic, and this summer I've been working with Dr. Claire Bates-Congdon at Bowdoin College as well as here. And I looked into a computational way of looking at co-regulatory factors and learning more about them during mesangial cell development. So, human genome is about 3 billion bases long, and out of those, only 3% are coding sequences, while 97 do not code for any proteins, but that's what we work with in our lab. And mostly because they play a key role in transcriptional and translational regulation of the coding sequences. So basically, Dr. Leif Oxford in MMCRI, Maine Medical Center Research Institute, have been working with embryonic development of kidney cells. And he has been very curious about the cells the cells of my interest, which are in the kidneys, then in the nephrons, the functional and structural unit of the kidneys, and inside the kidneys, we have the glomerulus, which is the key area where the filtration occurs from the blood. And inside the glomerulus, we have our cells of our interest, the mesangial cells. Basically, these cells clear out the debris and the unneeded residues so that the filtration can occur more easily in the later parts. But if something goes wrong and these residues are not cleared, it can result to chronic kidney failures, which has a prevalence of about 8 to 16% worldwide. So basically, in the mesangial cells, till now, we have 172 genes that have been identified by this paper by Brunskill and Potter in 2012. However, in this heat map of 7 different renal cells, we can see that majority of the 172 cells are also present in the other renal cells. However, Dr. Leif Oxford has been, went through this paper and came up with the 7 of these genes which are only expressed in the mesangial cells. And out of these 7, ETS1 is the gene of my interest, which is a transcription factor. So, goals of this project, it was a huge project. So I have 2 facets of my project, and the first project is to identify candidate cis-regulatory modules, the CRMs, of ETS1 regulation, while the second goal, or the second project, is to explore possible co-regulatory elements in the upstream regions of the 7 uniquely expressed mesangial genes. So you will come across this word a lot, CRM, in the later half of the presentation. So in our genes, there are sequences that we call, group of sequences that are potential transcription factor binding sites. And these are called motifs computationally. But these the transcription factor binding sites or motifs usually don't work alone. And they work as a unit that we call CRMs. Now, methods and finding, again, divided according to my first and the second project. For the first one, we get our rawest data is basically this huge sequence that we get from NCBI. And here, my raw data is the ETS1 intronic data, and intronic plus the upstream. And then in terms of processing, we use computational tools. And the first one we have is that GAMI CRM here, which was developed by the Congdon lab. And what it does, it finds the well-conserved motifs over evolutionary time and then assembles them into CRMs. And we feed this into what we call UCSC genome browser. And I'm so sorry for this huge, dense image. And it has a lot of information. But let me just give you what information you can get from this. Basically, it can show us where the gene of our interest is in a chromosome and also the scale. and then the gene of our interest, ETS1, and the tracks that I'm interested in. There are a lot of different information you can get from this, but the tracks that I'm interested in is the histone modification tracks, where the gene is highly susceptible to histone modifications, and where the DNase enzyme is most likely to occur. And last one is where transcription factor binding sites and where it's more likely that we can find transcription factors. And these are our output that we got from GAMI, which are human annotations. Basically, they mean genomic information that will tell us more about the gene that we're interested in. And I made a dummy of this huge diagram so that people won't get intimidated. And the first one, like I said, the histone modifications, the peaks, and then the DNase clusters, and the transcription factor ChIP-seq data. And all these three data are from the ENCODE, which literally stands for Encyclopedia for DNA Elements. And so these are the input data that we feed into the UCSC genome browser. There are motifs and our CRMs, and you can get a lot of CRMs, but which one are better than the other is basically the CRMs that are better will fit into this kind of grid, but it does not happen usually. And for results of this first project, I came across CRM6, which was 327 base pairs long, and they it co-occurs in the valleys and where the DNase is there, and also the transcription factor ChIP-seq data is found. And we found another one. There you go. CRM3, which is 380 base pairs long, and two more. So basically, transcription factor binding sites are normally the candidates, the cis-regulatory modules, are mostly about 200 base pairs to 700, 1,000 base pairs long. And so from this first project, we found 4. And now moving on to the second part. This is a very classic transcription factor binding to your upstream, non-coding part, which is the darker part, and then the gene gets transcripted and translated into proteins. So my question is to explore common co-regulatory elements in the 7 uniquely expressed genes, mesangial genes. But the question is, can we find a candidate transcription factor binding site that is common to all 7 of them? So the workflow for this one is very similar to the last one, but we got the DNA sequence from all 7 genes and added 2 more tools to our processing part, where the Summer 16 tool we just developed specifically for this project, and it identifies common motifs among 2 different sequences, and it's pretty much like comparing 2 different strands. And then Transfact is a software where you can look for known transcription factor binding sites. And the results from the first one, so we fed all the 7 in pairs and then into the tool. And we found, this is a Venn diagram of all the 7 genes, and we found 2 motifs in common among all 7 of them, which were highly conserved across more than 20 mammals. So the summary of the first one, basically we have 4 candidate CRMs, which are ready for in vivo work in a wet lab. And the second project gave us 2 highly conserved common candidate transcription factor binding sites in all 7 genes. And this makes us wonder if all these 7 genes are co-regulated together. So future work, like I said, the in vivo lab work. And second is to search for what transcription factor could bind to the 2 motifs. And we are trying to use Transfact and other tools, maybe develop new ones for this. And the third one is, so this was a third project, but basically we came across a colleague in Duke University, and she already knew ETS1 binding. She had a consensus ETS1 binding site, and which was an 8-mer compared to the 10-mers that we had. And these were highly conserved in all of our 7 genes. And we would like to ask more questions to this, saying if ETS1 is a putative co-regulatory transcription factor among all 7 genes. So basically, if we knew more about these 7 genes, we can possibly help mesangial cells regenerate itself without going through kidney dialysis or even transplantation, saving a lot of money and effort. Thank you, Congdon Lab, and Dr. Lee Foxberg and Nin Cheng from Duke University, and the entire of Congdon Lab, amazing people. And my funding specifically was from Main and Brief and NIH. And the Congdon Lab was supported by the Dartmouth Quantitative Biology COBRE and the NIH grant. Thank you. And these are the references.
Questions for Porcia?
Yes?
You said you'd like to see the CRMs that fall in the valleys of the histone modification.
Yes.
Is that histone modification, is that acetylation, methylation? Are there other?
So the peaks are where acetylation occurs the most. This part. This part? Would this work? Yeah. So basically the peaks are where acetylation is happening more, and the valleys are where it's the least.
And is that the only variable tracked on that line, or is methylation and other histone modifications also involved?
What do you mean by the only variable?
So acetylation, whether it's higher or lower, that's kind of the peak and
Yeah.
valley, right?
So the thing is, the UCSC genome browser has a lot of tracks, and there are methylation tracks too, but these are much more clearer for my project. That's why we use this one.
Okay. Any questions? Susan?
What is the source of your mesangial cells? Are they mammalian, and if so, which animal?
So we are a dry lab, so we do not touch any chemicals or any organisms. But basically, in terms of, to ask your question, the sources of my genes are basically mammals, only mammals, and it differs from, we get sequences of humans, of chimpanzees, bonobos, and all sorts of different mammals that are accessible in the NCBI genome database. Yes, sir.
Once you've done this bioinformatic approach and you get this analysis based on what you've selected to find your CRMs, is there a way that you can then validate biologically that this is a valid or accurate model?
So to answer your question, yes. And in terms of, first we find these CRMs. And then just because they are conserved over different species, divergent species, it makes sense that they have more more functional purpose. So we, in the lab, so out of these 7 genes, PDGFR beta has gone through the CRM process, and it's currently being validated in MMCRA in Dr. Leave's Oxford lab in mice. So basically, we find these computationally, filter them, and find the best ones we can. Because computationally, it says that, it means that we hypothesize that they have more, they are better than the others in more regulatory function. Yep.
Any more questions? And thank you very much, Porcia.
Yep.